मैं वही सोचता हूँ कि अभी तक मेरा ये कम क्यों नहीं होता है. डॉक्टर साहब कुछ सजेस्ट करो. हाउ कैन आई इम्प्रूव माई हेल्थ, राइट? सो दो आई रन फ्रॉम वन क्लास टू अनदर. सो व्हाट आई हैव डन इज दैट मैंने स्कूटी खरीद लिया. ये फेब्रुवरी स्कूटी आई बॉट एक्टिवा. मुझे लगता है मैं ही एकमात्र ऐसा फैकल्टी होगा जो स्कूटी से आई कीप ऑन रनिंग अराउंड राजेंद्र नगर वन प्लेस टू अनदर. ये चीज़ है. तो कई लोग ऑन द वे दे सी एंड दे स्माइल. आई आल्सो स्माइल. क्या कर सकते हैं, भागना होता है क्लास के लिए. एनीवे today's class will be important. Now see here what we have learned last class, that the things are there. See, we have covered this cloning part, right? And there I said that somatic cell nuclear transfer technique is one of the techniques which are used for this, right? Apart from this, I have also discussed about gene cloning. That is nothing but genetic engineering. That is called gene cloning. Just remember the technical word. See, whatever I'll cover in biotechnology was the topic. Today you will have notes also there. I have brought a few of the content which I'll give you the paragraph form also there. That will be sufficient. And then add current affairs. Like very soon, Vision IAS will have means 365. So that will be compilation of all the current affairs, right? So there things will come from the current perspective. So, when you read anything, let's say even science and technology, whatever topics we have covered, or you're coming in newspaper, then try to ask yourself that, what type of questions can be asked. Like today's Hindu paper, just the back page, right? Dragon, SpaceX. This article is there, right? SpaceX has got two type of launch vehicles. One is a Dragon, which is used for sending logistic to International Space Station. Second, you'll find Falcon, which is for the reusable launch vehicle type, RLV, right? There's one photograph given, HEHMS Queen Elizabeth. They're coming for that. It's the biggest warship in the England, named after Queen Elizabeth. And if you look at the photograph, it is an aircraft carrier. You see the dock is there. There is a sky jump. Right, that photograph has come in today's Hindu paper. At the back page, you'll find that. Right. Front page, you have one article, very important for, not from science and technology, the other area, that is a elephant census has come. You know, the data is 27,000 elephants and is alarming. Why? There's decline in the number of elephants. Now, what can be cause of that? So when you read article, then you should ask yourself, why there's a loss of elephants, right? What are the cause of elephants? Hathi mere sathi. So what a problem with hathi, right? Poaching. So, but poaching is been controlled. Most of time you'll find poaching activities have been controlled there. What is the prime cause of loss of elephants in India? Deforestation part. Now why I'm asking, because Odisha, Chhattisgarh, Jharkhand, right, and South India, Tamil Nadu, Kerala, Karnataka, and Northeast India, Assam, right, Meghalaya, Nagaland, that area, you'll find these are the critical places. Uttarakhand also, you'll find all these elephants there. So you should, what I want to just make you aware, when you read an article, right, whatever topic it may be, economy, environment, polity, governance, science and technology, you ask question there. What question can be asked in UPSC exam, in prelims form and the mains exam, right? Just it says that SpaceX has made continuous news. So that can be a part not asked directly. UPSC will not ask you direct question. Everybody reads newspaper, everybody knows what is given there. Something related to that question will be asked, right? Same way biotechnology, whatever I'm teaching the biotechnology and the material, so I'm compiling the material. The one thing is I'm putting on whiteboard, second I'm giving small paragraph there. At the same time, you'll have also one copy uploaded, because today will be ending with the biotechnology going to health area, healthcare. So that part, what I'm providing, that will be your kind of one study material which you can revise before the exam. Because sometimes what happens is that before the exam you don't have enough of time to go to variety of sources and read a lot of material. At one place you require, and then add with the current aspect. Keep on adding the current aspect, and when you read current affairs, ask the basic concept, what is the fundamental concept, what is application, what is government program, that part, which I've said very first class there. Like here cloning, I have given there. Nobody knew that the question may come on cloning, but question was asked in the exam. So whatever we are covering in the classroom, it is a, UPSC can go any of the area. You have to be totally done with your homework, that part. Then what I discussed last time, stem cells. Again, it is making headlines, continues for the last 6-7 years. 2012, there was a question on part of this. Now let me take you to the question there. Okay, what exactly biotechnology question is all about? Then I will start with the topic. Today's class will learn about again important topics there, like into bioremediation, gene therapy, right? Then we'll come to immunity part, immune function, and we'll learn about antibody producing mechanism called hybridoma technology. Again, the few topics will come. But let me just take you to the question paper there, PT question paper. Day-to-day basis means I feel that. See, remember that it's good if you can have time. But again, see, there's a way to read newspaper. When you look at newspaper, it's not that each and every article you have to read. First, you filter out, right? But yes, track it. Why tracking is very important. May not that today you may get time to read that article, but maybe that later on you know that this is the article and a question, if in case, comes in the exam, right? So that is always I advise that you have current affair material from Vision IAS and all things, classroom and everything. But please make sure if you have time, go to the newspaper. At least the Hindu, you'd follow that. No, science and technology doesn't come every day. It will come on an interval there.
Suppose there has been care of elephant. It has been coming in newspaper for past 2 days, but we have been, I have been ignoring it, being it a state issue that it is the state.
Correct.
So now it has come from the perspective of environment.
Right.
So shall I need to go back to those news?
Yes, that's again he has asked a very good question. Anything what you read there, you need to connect to the back. But something, again remember that what you have there in the earlier, if you can connect well and good, that is a very important thing which requires in UPSC preparation. Apart from that, you have to go to periphery. I'll tell you what is the periphery. Project Elephant 91-92, Government of India started. Objectives are given there, right? Now what comes there? Which law is there? Ask yourself that, what law? Wildlife Protection Act 1972, right? Then you ask yourself that, Elephant Reserve, what is the Elephant Reserve? Why one needs to have Elephant Reserve? What is the objective? Wildlife corridor, looking at the elephant problem. Then you ask yourself, how many elephant reserves are there in the country? Right? There is a factual part, whether 35, 40, 50. So that fact you have to, but though in the UPSC will not give you numbers. If 50 elephant reserve or 2 or 10, that will not be there. Then what is there? When you read newspaper, maybe later on, something or in that same article, a name of elephant reserve might be given. So, which state? What is geographical setting? So that is what you have to, again go to periphery, many things, which may not be in the newspaper. That's why UPSC exam has become very challenging. Because they are not asking very obvious kind of thing, something which is not obvious there. And that's why it requires a lot of hard work. To particular current affairs is a very challenging task. What to read, where you have to restrict yourself. When you go to website will have hundreds of things. And if you start reading one website with another website, third website, fourth website, you get lost in that. So much of information is there. So again, you have to control yourself. Yes, from UPSC syllabus point of view, this is what I need to know. I need not have to go to PhD research, right? That part of that, right? The basic thing of that. You're right. You have to connect the things.
Do science and tech, there is separate segment coming on Sunday. So
Yes.
Number of developments either by one of the institutes developing. So do we need to track that also?
Yes, you have to track. Sunday. Sometimes I always keep on saying what government programs, achievements part. So there you have to read that which scientific. Like yesterday, I've said CDFD, Center for DNA Fingerprinting and Diagnosis, right? There's one way could be a question in the exam that you have given the name of centers and which research area, right? Let's match the following, which is correctly matched pair. That can be one thing. Or when you read that Sunday paper, what happens? It gives you analytical perspective, mains exam writing, because you are able to connect with diseases, or let's say any kind of new, let's say biotechnology. We have discussed CRISPR. So there it gives you the ethical issues, social aspect, economic aspect. So that helps in mains exam there. PT exam will be direct. It will ask you that what is the concept of CRISPR? What is Cas9 and something, like different way question can be asked. Right? So again, very important. Sunday.
The problem comes is that it is technical.
Then when it comes technical, now you say that. Many of the students do not come from science background. or we would have studied science long way back, class 10th standard. After that, are not followed science there. And there's some keyword come, technical word comes, which you are not aware of that. Let's say RNA word has come, ribonucleic acid, or DNA, deoxyribonucleic acid. So there what you can do, underline those kind of keyword where you think that you are not comfortable. Ki what is DNA, what is RNA, and there comes single guide RNA, sgRNA, what is there. So first question comes, what is RNA? Then you have to go back to the textbook to learn about that particular term, so that you are able to build up the knowledge. Like for example, RNA word comes. RNA and DNA both are called nucleic acids, a type of biomolecule. Right? DNA is double-stranded molecule, where RNA is single-stranded molecule. RNA is called ribonucleic acid, ribose sugar, 5-carbon sugar, and the nucleotide. What are nucleotides then? AUCG, adenine, uracil, cytosine, guanine. These are the building blocks of. On the other hand, DNA is double-strand, complementary. It is made up of deoxyribose, that is sugar, carbohydrate, 5-carbon sugar. Then you have nucleotide, the nitrogen base, ATCG, adenine, thymine, cytosine, guanine. This is what biology. What I'm saying, this is the biology. Right? So that biological aspect, you should be aware that what is DNA, what is chromosome. Or let's say I have used several times in the biotechnology word gene. Right? So what is gene? Gene is the information on DNA, which codes for a trait, character. Right? Or maybe for a protein. Hemoglobin, myoglobin, insulin. That information on a DNA is called a gene. That's what. So basic keyword when it comes. Let's say we have used the word launch vehicle. So there I've explained in the classroom. ELV, RLV, expendable launch vehicle, reusable launch vehicle. Missiles. Again the word ballistic, cruise, tactical, theoretical, those kind of things. And I'm very sure the moment in the science and technology, the slowly when you keep on learning and developing things, it will become very easy for you. And at the same time, if something very advanced comes from nanotechnology, robotics, material science, don't worry about it. Don't panic in that case. Just try to find out and try to learn. But again, if it's very technical, leave it. Right? You should know that something linked with that, but don't go into that entire engineering aspect or scientific, because that part will not be ours. But common word, particular useful word, that you have to be aware. Right? I'll give you example. You'll just understand with the example what we have learned in the classroom. That's what I'm spending 10-15 minutes extra time and going to that. Let's say here 2012 paper. This is what I have opened now, right? And I'll come to that stem cell question there. Now, question 16. Everybody can read this? With reference to stem cells, frequently in the news, which of the following statements is are correct? Right now, number one, stem cells can be derived from mammals only. This is how the question comes into that. Now, what is given? Stem cells can be used for screening new drugs. And number third, stem cells can be used for medical therapies. Right? This is the way UPSC asks question there. Now, if you look at this question there, you have been given option 1 and 2, 2 and 3, 3 only, and 1 to 3.
1 to 3.
Statement number 1 is correct or wrong? How many of you think that statement number 1 is correct? Okay. Those who think wrong, why it is wrong? Okay, now that's what I said now. UPSC question language will be like that. It asks the question there, concept part of it. Now, what I've said in the class, see, remember. Even if in such question in the exam, you are not aware whether mammals, reptiles, birds, or where it is there. Normally in newspaper, what do you read? Mammals only. Whether chimpanzee or apes or humans, that only comes. Very rarely you come across bird or reptiles there. Now look at it. But how you link that? I'm giving you a smart approach. What is smart approach? I've said in the classroom that stem cells are two types. Embryonic and adult. So what is the key word here? Embryonic. Any species which grows through embryo. Because stem cells are what? Undifferentiated cells. So that means bird or let's say reptile. Right? Reptile also will have embryo.
No, but still lay egg.
No, but they lay eggs there. But even the egg also there. There's something on that particular species related stem cells. That related stem cell will be there. Like bird will also have there in the part of that. Right? Amphibians will also. He's right there. It is not like a uterus or something fetus there. But still remember that they have their tailored for that particular way there. Because what are stem cells? These are the progenitor cells. They give rise to all type of cells of the body there. That is the thing there. So first statement is wrong because the only word is there. The only word. So remember that in living world, animal kingdom, the stem cells are found in many other organisms. Right? Not only the mammals, it will be also in reptiles, amphibians, and even in the case of the birds also. That also comes into that. Second is correct. Diseases and toxicity. Third is also correct. Regenerative medicine. So here answer is 2 and 3. So remember why challenging it becomes. Something you will be aware, something you will not be aware. And something where you are not aware, then you have to, goes into fix. Whether I should take a risk or not. And you have the option 1, 2, 3 also there. Right? So इसलिए यहाँ पर ये जरूरी होता है. Right? Now this part, and after 2012, अभी तो question नहीं आया. So let's say 2018 exam. You come across something on IPSC, induced pluripotent stem cell, Shenia Yamanaka genes, and that part. So at least now you know that sometimes it is possible that we can insert from outside foreign gene and convert any body cell into stem cell. Right? And then we can produce any other type of body cells. So that concept you know that. And इसी चीज को वो घुमा फिरा कर पूछेंगे. Right? That is the way out. Now look at here. This question number 18. अभी मैं आपको एक और तरीका में देता हूँ. Question 18 there, the other side. This has gone that side, but I'll read the question. A team of scientists at Brookhaven National Laboratory, including those from India, created the heaviest antimatter, anti-helium nucleus. What is or are the implications of the creation of antimatter? Number 1, it will make mineral prospecting and oil exploration easier and cheaper. Number 2, it will help probe the possibility of the existence of stars and galaxies made of antimatter. And third, it will help understand the evolution of the universe. Second and third, you are very right. Right, because antimatter, anti-hydrogen, anti-helium, anti-carbon, nitrogen. Now what is the theory says? That when the universe was born, Big Bang. So why don't we find the galaxies made of antimatter? When there is a matter, the opposite of the antimatter. What is antimatter? Are those which consist of antiparticles. What are antiparticles? Which are same as particle but differ in charge and quantum properties there. Like positron, electron. Electron negatively charged, positron positively charged. Size is same, mass is also same. So why don't we in universe, we come across a galaxy made of anti-hydrogen, anti-helium. So that is what the scientists tried to do that time. That is the current affairs news. Again remember that. 2012, we have opened the question with 2012. So that is related to development 2011-12. No, not parallel. That is parallel. It's our universe only. The universe where we live, based on that. The universe only there. Composition of the universe. Why don't we find those kind of antimatter there? The 2 and 3. Answers 2 and 3. Right, that part comes. Now if you look at here, see. Question number 19. Which of the following is are cited by the scientists as evidence or evidences for the continued expansion of universe? The number 1, first, detection of microwaves in space. Number 2, observation of redshift phenomena in space. And third, movement of asteroids in space. And fourth, occurrence of supernova explosion in space. Now see, this question uses number of technical words. And this one, not one, 4 are there. That means you have to know the information from 4 areas. Then you can decide whether that can cite evidence for expansion of universe. Now why 2012 this question came? Because 2012 the Nobel Prize was based on the supernova chasers. Those scientists who studied supernova. What is supernova? Exploding stars in the universe. and UPSC asked the question there. Again linked with what? Current aspect. But what again remember that? Here the question asking on the basic concepts. Now tell me first, common sense, first statement, correct or wrong? Why first is correct? Universe is expanding, okay? Now remember that. Here is what expansion of universe is asked, right? This is the part has been asked, expansion of the universe. The evidence for this was the question asked. Right now let's look at here. How do you feel that something is like this universe is expanding? What is one way out?
Distance.
If distance increases, things move away from each other. That's only the expansion. If the space expand, then things are moving apart from each other. So that is one thing what called Doppler's effect. There is a concept called Doppler's effect, and Doppler's effect is observed both in sound and light also. There is one option also given there. What is Doppler's effect in sound? Which you know that if any object which blows horn or emits sound and comes near to us, it appears that it's louder. But actually it is not louder. Frequency is the same. If any object blowing the horn or let's say vehicle, car or train moves away, so what do you find that? It is decreasing there, but it's actually not decreasing. Frequency remains the same. So that we perceive as an apparent change. Same happens with light also. So it is like this one. If any object, let's say, and this is a, let's say, things, let's say Earth, and you have, let's say, star, some star is there. If there is expansion, so they will move apart. They change in distance. So what happens in sound? If anything goes away, we hear lesser. Intensity decreases. So what is decreasing there? Frequency decreases. This is what you have to link with general science concept. That decreases. Now from general science, from physics it comes, frequency and wavelengths are inversely proportional. So that means wavelength is increasing. If wavelength is increasing, it means that if you look at this part, Vibgyor, light is made of this spectrum. This is called blue end and this is called red end of spectrum. So where is the longer wavelength? Right. So that means whatever the observation lines are there, they'll move towards the red side. This is what called redshift. This is the concept being asked. See there, you have to think of that. So that means second is correct. Expansion of universe means there is a redshift phenomenon happening there. Now coming to the first one, detection of microwave in space. There are a lot of microwaves. The universe is expanding. Okay, that's part. Now what the third one? Movement asteroid in space. Where do you find asteroids? Between Mars and Jupiter. Can we find out there's something expansion happening? No, you cannot find. The third is ruled out. Now what the fourth one? Occurrence of supernova. It will happen there. This is what. So what will be answer here? A or B? But anyone want to object the first statement? Microwave. See, microwave is what? It's a kind of what we use radio waves or something of that. Can we find out distance change?
Yeah, by sending and receiving the microwave the time.
Hardly, but it's a detection of microwaves in space. Yes, that will. That would say sometimes the statement will not be very clear. Right? That's again that becomes a difficult. Right? So here the answer 1 and 2. That's what I'm saying. So but what is said here? Are you aware of this concept? That's the thing there. If you are aware of this one, you know that next answer could be of this kind of choice. Now I'll take another one. Then I'll start with the topic of today's class. It's a stem cell, I have said that. Now this one. I'll take a biotechnology part. One and two there, answer one. Answer 1, 2. Then now I'm coming to this one. Very interesting question was there. I just wanted to go into that part. All the science tech question will find 3, 4 questions together. They'll come together there. Now this question 65. Question 65. I'll read out there. People who cannot read from this side. Other than resistance to pests, what are the prospects for which genetically engineered plants have been created? Number one, to enable them to withstand drought. Number two, to increase the nutritive value of the produce. Number third, to enable them to grow and do photosynthesis in spaceships and space stations. Fourth, to increase their shelf life. And the options are there. Let me just. You'll find that 1 and 2. I'll just make it smaller. You have 1 and 2, 3 and 4, 1, 2, 4, 1, 2, 3, 4. Like you now see what is saying that now we're trying to grow it to the special. But what is the thing? This question came 2012. You have to look from that context what was there in the current aspect. Right? So that time the answer is 1, 2, 4. Like, though the scientists are trying to experiment whether we can grow artificially in the lab, in the spaceship or space station in terms of photosynthesis. But today the benefits of a lot of things have been developed there. Right? This is part of that. So biotechnology question, what I want to say, biotechnology question will not be difficult. Basic terminology will be there. It will be used in that case, right? So that is part. Now let's come to the concept part. So what I'll do, I'll take whatever I explain, I'll take with the question also there. So now let's understand. Yesterday, whatever we have learned, DNA fingerprinting and all. Now I'm coming to one concept and then I'll come back to the biotechnology method for that. It's again a very important thing. So what we are going to learn a technique that is the hybridoma technology. Hybridoma technology. But before I go into the hybridoma technology, so for the same stem cell will be same. But let's say the stem cell from some other cells, it will have different DNA will be there. Different DNA will be different from there. Other cells will be same. Like what was the skin cell? All skin cell will have the same DNA. Like because all the body cells, somatic cells, they have the same DNA. In the RBC now again here. What is asking? RBC doesn't contain nucleus. Red blood cells doesn't have DNA. But now the question is that blood is used as sample for DNA fingerprinting. Getting this point? What is asking that? RBC doesn't contain nucleus. There is no nucleus, so there will be no chromosome DNA. But in the testing for DNA fingerprinting, one can take blood sample also. So other cells here, like white blood cells. RBC itself will not give any DNA fingerprinting. The other like leukocyte and like in the case of WBC, white blood cells, they contain the certain type of WBC which contain nucleus. So from there DNA can be extracted. That is there. But if comes RBC will not have. Because RBC doesn't have nucleus, then no DNA will be there. That part. So blood other components, not RBC. Not RBC. Okay, now before I go, let me go to a very important concept, immunity. You will understand hybridoma only when you understand the concept of immunity and in terms of antigen antibodies. Then we will come to that part. Now, what is immunity? Is a defensive mechanism. Right? Immune system, this immunity is related to defense mechanism. Defense mechanism of any organism, whether you talk about bacteria or fungi or plants or animals, every living organism has got immunity. Defense system is there. Right? Even humans have got immune system there. Our immune system works with a special type of cells, what is called lymphocytes. But before that one, let's come to that. How many types of immunities are there? The concept of immunity, how many types are there? I'll use this word here, active and passive. So I'm just going to the biological part, then I'll come to the biotechnology methods. Now, active immunity and passive immunity. As the human beings have got two types of immunity, active immunity and passive immunity. Now, what is active immunity? Where immune system, right, is activated to produce antibodies against the invading pathogen. When bacteria or virus attacks or goes inside the body, so our immune system, which is which works with WBC, white blood cells, recognize the antigen on the bacteria, virus, and produce antibodies. So what I want to write simply here is that, the immune system is activated, that's what the active comes, is activated to produce antibodies. So that the WBC is working to produce antibodies. Now what is passive immunity? Our immune system is not activated, but we take it from outside. Antibodies comes from outside. So here what happens there? The passive word means here that immune system is not activated to produce antibodies, but works with antibodies from outside. But works with, I'll keep on reading so that people who are not able to see there, works with antibodies coming from outside, from another individual, but is from outside. What I've written on the board? Immune system is not activated to produce this. That means WBC will not produce. And we get ready-made antibodies from outside. That is a produce antibodies, but works with antibodies from outside. Right? Now, when you look at active and passive, there's a two terms which are explaining there, right? Now, there's another term you come with, innate. The word innate comes, and another word comes, acquired. Innate immunity is based on from birth, which we get from there. Acquired, we acquired during our lifetime. Even after birth, we can also acquire the immunity part of antibodies can produce. And that acquired immunity can be both active and passive. What I want to say? When you talk about acquired immunity, that acquired immunity and innate immunity can be both in that context. Now, I'll tell you that active immunities are not fast, it is slow. Whereas passive immunity is fast. Very clear to the concept. Why active immunity is not fast? So that's what remember that. Yeah, active का मतलब ये नहीं कि action. Right, action part is not there. What I've said, WBC is able to produce or not. That is part. ये slow क्यों होगा? It will take time to produce antibodies. WBC will take time to develop those antibodies. But here what happens? You're taking antibodies from outside. So those antibodies are present and that will fight against bacteria and virus. So this is slow, but this is fast. This works in a faster way there. Now come to innate and acquired. Now, innate from birth. From birth what happens is that innate activity. Now when you talk about one aspect which comes there after birth, the newborn baby gets the antibodies from mother's milk. Right, that's the acquired part. And that is what called passive. Because mother's milk contains antibodies and that gets transferred to the baby there. So that is a passive kind of thing. But let's say a baby develops an infection. Let's say viral flu or something. And when the baby is cured of the disease or that problem, so by that time WBC has learned and then produced antibodies. So next time they will not be there, right? Now vaccine, when you look at vaccine, the vaccine comes in this part. Right? Where we come to vaccines? Vaccine is based on the part of this part, the active part. Because when you take vaccine, what does vaccine contain? So vaccine is basically anti-sera. Right? Vaccine is what? Is a serum. Blood has got, when you remove all the parts, WBC, RBC, platelets, it forms a sera. So anti-serum is sera containing antibodies. Right? Anti-sera, right, which is serum, blood serum, which contain antibodies against pathogen. So what is vaccine? Vaccine is anti-sera made up of, what I'm using the word, which you all know that, attenuated or killed pathogens. Let's say bacteria or virus, they are not live form, they are dead form. Attenuated. So what I've written? What is vaccine? Vaccine is anti-sera made up of attenuated or killed pathogen. Active and passive. See, passive could also be there. I'll come to that. First to understand active one. Vaccine there. So what happens in vaccine? Let's say when you say hepatitis vaccine, it contains that hepatitis virus. Let's say when it's a polio vaccine, the polio virus, but it's not live polio, it's a dead polio. So what happens? Our immune system, WBC, white blood cell, recognize that antigen on the polio virus and makes what? Antibodies. So that is what called active. But again remember, there are vaccines which contain antibodies also. So what I'm writing here, vaccines are not only this anti-sera, which is made up of anti-attenuated or dead viruses or bacteria, pathogens, but it can also be anti-sera containing antibodies. It can also be containing antibodies. That means vaccine can be both active and passive. ये बहुत इंपॉर्टेंट कंसेप्ट ध्यान रखना है. लेकिन जब पैसिव की बात हो रहा है तो उसमें क्या है, एंटीबॉडी हमारे WBC नहीं प्रोड्यूस कर रहा है, हम बाहर से ले रहे हैं. उस वैक्सीन में वो एंटीबॉडीज है. कैन यू गिव एनी एग्जांपल ऑफ दिस? सी मोस्ट ऑफ द वैक्सीन इज दिस वन. पोलियो, हेपेटाइटिस, all these measles are this side. But I am saying that there are vaccines which may contain antibodies from animal, maybe from animal and transfer to humans. Dr. Sir, for example. Yes, yes. Snake venom, doctor. Venom. Right, snake bite and all this. वहाँ भी है. What about this diphtheria, pertussis, tetanus? Like tetanus. What is tetanus there? It's a toxoid. Tetanus injection what you given, the toxoid is there. It's not contain any antigen. You know that sometimes horse is immunized and horse produce that antibody, and then from horse blood that is taken and extracted given to humans. Right? This is what I said in terms of sometimes you'll find vaccines which are from the animal origin. ठीक है, तो आप mostly ये याद रखना है, most of the vaccines are active only because we take bacteria or virus which are in dead form, and our body produce the antibody in response to that. ठीक है, एक तो ये चीज है. Attenuated means they are killed in that way. That means destroyed, so that they do not work. They do not remain active, inactivated form, destroyed in that form. So they will not work. Now I am coming to that part of this. Now what I am coming here, biotechnology today has come out to a stage where we can produce artificial antibodies outside in the lab without producing human body or animals. That's why I've written the name hybridoma technology. This I've given the concept. What is immunity? Immunity goes through the antibodies, and in that case, vaccine comes. But vaccines are generally in this form. But let's say we require antibodies in large quantity in the lab. Every time we cannot put the injection antigen to the body and develop antibodies there. That's good. Or to any animal, lab testing. So that will be again not feasible. But biotechnology gives an idea, gives a method by which we can produce a large quantity. And there that method is called hybridoma technology. So what I'll do now, after this giving the biological concept, active, passive and vaccine and this part, I'm now coming to the biotechnology method of producing antibodies in the lab. Outside human body or any animal, we can produce that. Now how you understand the term also, the meaning of the word also there. And then I'll give you paragraph writing there. But first let me draw a diagram to explain what is hybridoma technology. Right, now understand this way. Hybridoma technology is basically first time, so I'll give you the name of the scientist also. This is a Caesar and Milstein. These two scientists, right, first time in the lab tried out this method to make artificial antibodies or synthetic antibodies. And they were awarded Nobel Prize in 1984 for doing this work. So what they did was that developed this method. They have developed this technique there. The technique was developed by them, right? So that's what every top technique I am giving you certain name of scientists also there. Now understand what is the point here. What they did was that, what exactly they did? They took a chuha. So mouse is a favorite animals for the experiment. Right, the chuha was taken, healthy chuha, and this chuha was given injection. Antigen was given to it. Let's why antigen? Because this chuha has to make the antibody of this antigen. Right? Injected. Chuha was killed. Right? And then WBC was taken. Now what I said? Antibody producing cell is called lymphocyte. Right? So that is I'm writing here. here antibody producing cell, and I'm writing one very important name which is part of human anatomy also there. It's called spleen cell. Liver and spleen, human body also, we have spleen cell. And these are WBC containing kind of white blood cells hota hai usme. So that cell was taken. Let's say if I draw this one, this is the antibody producing cell.
Lymphocyte.
हाँ lymphocyte भी है, lymphocyte में WBC कहते हैं जो कि एंटीबॉडी बनाता है वहाँ पर, तो वो भी आप कर सकते, लेकिन मैंने सिंपल लैंग्वेज में एंटीबॉडी प्रोड्यूसिंग सेल लिख दिया, ठीक है, लेकिन लिम्फोसाइट होता है. और नाउ यू सी दैट. अब दूसरा क्या किया, दूसरा चूहा लिया गया, उसको थोड़ा पतला बनाता हूँ मैं, वेरी थिन एंड बेचारे लॉट ऑफ UV रेज, X-ray was passed, and this chuha became cancerous, developed cancer. So this is a cancerous mouse or mice, cancerous, right? And tumor cell was taken, tumor cell, cancer cell, right? It is called tumor cell there, and there is a name for tumor cell. What is called here myeloma. Right? Tumor cells are there, myelomas. They are called myeloma. Myelomas are there. What is the property of myeloma cell, cancer cell or tumor cell? Uncontrolled cell division. They multiply divide, they uncontrolled cell division. That is a part of it. Let's say this is a cell there. So it has what? Uncontrolled cell division. They'll keep on dividing there. That is the property of myeloma cell. Whereas the lymph cell here or antibody producing cell will produce what? Will produce antibodies. And what antibodies will be there of this antigen which the mouse was given? Produce antibodies. So what this scientist did was that they combined these cells together, cell fusion. Right? They combine the cells together. So what will happen there? When you combine this cell together, so you'll have a resultant cell. The cells were combined together, fused together. Right? Again, we don't have to know the protocol, what is done, but they tried level best and they combined both the cells together to form single cell. What is called hybrid cell, right? So what this became there? Hybrid of two cells, antibody producing cell and cancer cell. And since it is from myeloma, so he was said hybrid myeloma, and that is called as hybridoma. The word has come here, hybridoma. Now here, this cell will have both the property of parental cells. What is the property of that? It can produce antibody also, and it will also keep on dividing. So that means it will have division also continuously. At the same time, they will also give rise to antibodies. So now if we can culture this cell, so we will be able to generate antibodies in a large quantity there. So here, this hybridoma can be cultured, but there are different methods where is the culture there, can be cultured to produce what I'm writing, very important word here, monoclonal antibodies. There's a term called monoclonal antibodies. This is a technique. This is just you have to know that antibodies are produced in the lab, synthetic antibodies, monoclonal antibodies. What is the monoclonal? It's not that same type of process. This is one type of function. Clonal here means in terms of their functionality aspect. That part, epitope and all this concept. Right, this is what was done in the lab. So that we can produce in the lab antibodies and use it in various applications. Right, so this was tried out. So I'll put in a paragraph there. Then I'll come to application part, where we use this kind of method in a regular experiments and application part. I'll be coming to that one. Now remember that. Last time when I was taking the class, at that time I told in the classroom that cloning has not been asked. And the question came. So in this batch, I have said, till yet in last 10 years, the question has not come on this particular topic. So let's say in the exam, or any this hybridoma word comes, at least you know that this is the thing they have done. Right? This has to be at the back of the mind. What exactly hybridoma? But what is very important? Applications. Where the application of the thing comes in. Right? That part is there. In the lab, we do cancer cell, antibody producing cell combined together to have a single type of cell which can have both the functions together. That comes in. Right? So I'll give you the notes also there. I'll wait for that. You can have.
You said tetanus gives the passive immunity. What is the active immunity?
No, tetanus is that I've said toxoid. Active immunity only, but it is not the actual, you can say, pathogen there. That is part of it. It is active only, because if you do not have active, then it will be difficult. But we don't take the bacteria and put it there. That's part of it. I'll be coming to the health aspect also today's class. After these techniques are over, then I'll come to that part. So let me come to the written part of it. So I'll dictate from my textbook. DNA fingerprinting yesterday already I have given there. Okay, now right. Everybody has written, drawn this diagram. Now below that you write. Hybridomas are cells. Hybridomas are cells that have been engineered. Hybridomas are cells that have been engineered to produce. A desired antibody in large amounts. Desired antibody in large amounts. I've already written monoclonal antibodies. I'll not give that part. Desired amounts, give a full stop. Same paragraph right there. Monoclonal antibodies can be produced by hybridoma technology. You have to stop there. Now same paragraph you write. In this technique, antibody producing cell such as lymphocyte, lymphocyte, such a lymphocyte, and cancerous cell such as myeloma are fused together. Cancerous cell such as myeloma are fused, F-U-S-E-D, fused together to produce hybridoma. Hybridoma cells. Right? Give a subject, hybridoma cells. Now, I have already said that this Milstein and this Carl Milstein and this part, they have won Nobel Prize in 1984 for this work. This is only a biology part. This I want to give you that knowledge there. So that's what I'm asking question. I've used the word lymphocyte, WBC, white blood cells. A very GK type question, nothing to link with the biotechnology part. How many type of lymphocytes are there? Or how many type of WBCs are there? 5 types. Which are those? All right. इसका बहुत सिंपल तरीका याद करने का. I'll just give this. मैं इसको बताता हूँ. कभी भी अगर एग्जाम में क्वेश्चन आ जाए तो सिंपल तरीका है. Never let monkey eat banana. This is how I have remembered. Monkey eat banana. कभी भी बंदर को केला मत खाने दो. That's it. Never here means neutrophils. What I've given here? Neutrophils. Right? L here stands for lymphocytes. Lymph organ, and there is something called lymphocytes. M stands for monocytes. E stands for eosinophil. And B stands for basophils. Okay, these are 5 types of WBC, out of which lymphocyte becomes very important. This one. Right? दो तरह का बायोलॉजी में जब पढ़ेंगे इम्यूनिटी सिस्टम तो आप उसमें आप पाओगे कि टी लिम्फोसाइट, बी लिम्फोसाइट. टी सेल जो बात हो, टी सेल्स, थाइमस, टी स्टैंड फॉर थाइमस, तो वो आता है. आउट ऑफ दिस 5 WBC, all are involved with immune function, but the most important defense function comes from lymphocytes. That is what I've just given a way out. Never let monkey eat banana, right? Neutrophils, right? There's monocyte, lymphocyte, eosinophil, basophils. These are 5 types of them. Right? This is clear here? Now my question is that antibodies are used in vaccine program, develop vaccine. Now I am asking a very basic question. Which other are application of antibodies? Which all other things antibodies are used except vaccine development program, where all the antibodies are used, so that this technique will be useful. Can we put into that? Yes. Diagnosis. How diagnosis can be carried out? Blood transfusion. Okay. Apart from that, why I ask this question? Because there is something I wanted to explain here. That's what I brought there. There's again biotechnology comes into that. See, let me write here. If you look at this application part, the first application of this technique will come into vaccine development program. Second comes here diagnosis, medical diagnosis of disease, let's say HIV. What is done in HIV? How the person is detected, that person is positive or negative. So what type of test is done? ELISA test. That's what I wanted to. Like diagnosis. For example, ELISA. What is ELISA full form? Enzyme linked. Yes, it stands for enzyme linked immunosorbent assay. That's the full form of ELISA. Now what is done in ELISA? I'll come to that. In ELISA test, what happens is that you'll find that there are plastic wells are there, right? This kind of plastic wells you'll find, and this plastic well is coated with antigens, for which antibody has to detect it. So the plastic well, let's say, where you have the antigen is, remember that antigen antibody, they combine together, react together. Let's say HIV ELISA will have HIV antigen. What is HIV antigen? Protein on the HIV virus. That is the antigen. And let's say a person has got HIV virus, then the person's immune system will develop antibodies, immunoglobulins against that virus, HIV virus. So when you do the testing there, so let's say antibody coming from the blood, right? So they what happens? They can combine together, react together. And then what is done here? What enzyme? Certain enzymes are there which can give color. Color is developed. Luminescence part. So if a color is developed only when antigen antibodies are reacted together, then that enzyme works and gives the color. So that means positive person has got the virus, right? If the color is not developed, that means reaction is not there. That means the person is not having that antibody. That's why antigens is not be able to capture that antibody. Reaction is incomplete, so it will give negative result. This part is the normal. So whether you talk about HIV ELISA or hepatitis ELISA, all the ELISA test works in this way. So what do you require? This one is antigen to develop this ELISA kit. But sometimes ELISA could be this also, that antibody will be coated. There will be coating of antibody which we cannot see, which coating is there. And that captures antigen of the virus present in the body. That part. So let's say, for example, dengue. Just take example, dengue, chikungunya, swine flu, right? Dengue is a virus, chikungunya is also virus, swine flu is also virus. The virus will have what? Protein, that will have antigen, and that is captured there. There's antigen from the blood, that is coming from blood, that will be captured there. Now tell me, out of these two, which is more sensitive, which will be more better to give the result? Which will be? See what I'm saying? ELISA can be two types. One ELISA is that where you have coating of antigen and that captures the antibody from the blood. Second type could be what? You have the coating of antibodies and that captures the antigen of the bacteria or virus in the human body. That is coming from the blood. Right? So which will be more better in both? Second one or first one? Second one. Why second one? Because remember that here false positive result can be result. When you talk, that's ELISA test. Normally ELISA test is what? This type. Antigen is coated and that can give false positive. Imagine HIV case, right? HIV AIDS. you don't have the virus, and ELISA test comes positive. Then you will start asking question. ये क्या हो गया? You start exploring all the options. How can I become positive? And then you say no, नहीं हो सकता है. I cannot be positive. ऐसे भी कोई positive बता दी और चाहिए positive, half of you're gone. आधी सांस तो वहीं अटक जाएगी. But let's say you take that himmat and you say that okay, no, this cannot be there. Something wrong is there. So something wrong is there. Where wrong is there in this test, ELISA test, because some other antibody can also react to give the positive result, which may not be for the HIV. There sometimes happen there. This kind of result also comes. So there, in what case the doctors do or hospitals do there? क्या करते हैं डॉक्टर साहब? अगर ऐसा false positive आ गया तो क्या करेंगे? हाँ, तो they go for confirmatory test. Another test is done. Really it is a positive or negative. That is what is happening there. So what I want to say, let's say this type of ELISA, you require to have antibodies, and how you can make it? Hybridoma. That's what I wanted to say there. I've given you all this information only to just to make understand. To have more better ELISA test, which is antibody coated, so how would you make that antibody? You cannot inject a person with HIV virus and make antibody. That is not the way out. Or any animal. So what is the best way? Going for hybridoma. In the lab you can develop. And then can go for making an ELISA kit for dengue or chikungunya or any kind of thing there. So that's what I've said. One of the applications, vaccine development, second come diagnosis. Now third, very important, therapy. And this hybridoma technology is also useful for therapeutic purpose, treatment, therapy. And I'm giving example, cancer. Certain type of cancer can be treated with the help of hybridoma technology. Can you think of कैसे काम करेगा? How it will work? If we want to treat cancer with the help of antibodies, this type of antibodies which will be used for treatment of cancer. So how it can be done? Just think of that, how it can be done. Therapy part, I've said. We can also use antibodies, monoclonal antibodies for therapeutic purpose, for treatment part. Okay. What we do in cancer case? क्या होता है cancer case? I have said in the classroom, radiation. We try to destroy the cancer cell. That part of that. What is another way of doing that? Let's say drug delivery. कैंसर को दो तरह से किया जाता है. One way is that you use gamma rays, right, radiotherapy, and kill the cell, cancer cell. Second is what? You deliver the drug, right, chemotherapy. You deliver the drug to the cancer cell, which can act on the cancer cell and destroy the cancer cell. So that comes what? Targeted drug delivery. So I'm coming here, for example here, targeted drug delivery. Targeted drug delivery to tumor cells can be done through monoclonal antibodies. And I'll explain with a diagram how it can be possible. We make in the lab this antibody. What I've drawn in the diagram, this is what antibody looks like, right? And what we can do, we can tag with the anti-cancer drug. These also scientists have done that. Now what will happen? This antibody will go and bind to the cell for which the antibody is developed. So इसको जब हम use करेंगे तो क्या होगा? This will go to the cancer cell. This is cancer cell, and cancer cell contains this kind of antigen. And for this antigen, this antibody is there. So this will go and bind that one and deliver the anti-cancer drug. So there will not be side effect. There will not be any side effect because the drug is delivered only to the specific tumor cell. That's what is there. So what you have done, you have used the antibody for this particular cell, right, which goes and bind and deliver the drug to that. This is what I've said is another way. So what we can do, we can make artificially this antibody in the lab for the cancer and then tag with the anti-cancer drug and then use in the human body. That part is there. ठीक है, ये चीज़ है कि हम ऐसा एंटीबॉडी लैब में बना दे, उसमें एंटी कैंसर ड्रग दे देते हैं. तो जब वो एंटीबॉडी जाएगा वो सेल पे, ट्यूमर सेल पे डिलीवर कर देगा. तो उसके लिए पहले उस ट्यूमर सेल का स्पेसिफिक एंटीजन को लेकर एंटीबॉडी तैयार करना होगा. देयर वी कैन यूज हाइब्रिडोमा. दैट मेथड कम्स इन. एंड दिस हैज बीन ट्राइड वर्ल्ड वाइड. दिस हैज बीन डन फॉर कैंसर ट्रीटमेंट एंड ऑल. इट हैज बीन ट्राइड आउट. राइट, इम्यूनोलॉजिकल रिसर्च इज ऑल पार्ट में आता है. नाउ यू कैन राइट समथिंग आई वांट टू गिव हियर.
But this is not in practice now.
Not in practice there, but this has been done for, can be testing part of that.
Chemotherapy.
Chemotherapy practice में, chemotherapy direct drug देते हैं. What happens? Chemotherapy directly is given. So that in that case, you know side effect. Person loses hair, color becomes, a lot of side effects happen. Why? Because you are using such kind of toxic kind of drug. And right. But this is one way where we can use monoclonal antibodies and go for a targeted kind of delivery.
Practice में क्यों use? Why is it?
Most costly है because you have to make antibodies, develop in the lab, then go for this one. So only a few of the cases where people can afford, they can go for such kind of advanced technique, advanced method. Right, that is what. Normal use is vaccine program and all this is used there. उसके लिए use करते हैं. एक और चीज में लिखा था, you can write such monoclonal antibodies. After that, such monoclonal antibodies have opened such monoclonal antibodies have opened remarkable new approaches to preventing diagnosing and treating disease give a full stop there. Same paragraph. Monoclonal antibodies are used to distinguish. Monoclonal antibodies are used to distinguish between B lymphocytes and T lymphocytes. Are used to distinguish between capital B dash lymphocytes, or B cells, you can write B lymphocytes and T lymphocytes. Again, if you want to purify, which one you want to, there again one can be used there. B lymphocytes and T lymphocytes. ये फर्स्ट शब्द है. और ये कहाँ पे हेल्प होगा? What HIV virus does? HIV virus destroys the T cells, their CD4 and all this. You can, after this last sentence, right? This knowledge is helpful. Same paragraph. This knowledge is helpful not only for basic research. This knowledge is helpful not only for basic research, but also for identifying different types of, and also for identifying different types of leukemias. LEUKEMIAS, leukemias. So what are leukemias? Cancer, blood cancers. Right? LEUKEMIAS, leukemias and lymphomas. LYMPHOMAS, lymphomas. I'll spell it. LYMPHOMAS, lymphomas. Lymphomas are again blood cancer or lymph cancer. Yes, that comes. They destroy the lymph node and all this part of cancer. Diseases are there. So that can be again used for kind of identification. So we'll give full stop there. So if any question comes, and this is what standard from what comes in generally biotechnology thing which I've given there. So let's see that. Agar aa jayega to well and good. At least that part I've covered. Right? Now I'm coming to another thing where question came in this year exam. But first of all, let me take the question there, and then I'll come to the part of it. Monoclonal means those antibodies which are for one subtype, one type of function there. See what happens when our body produces different type of antibodies. And in biology they are called immunoglobulins. 5 types are there. So when you say monoclonal means a population of same type, which is for that particular antigen. That is what called monoclonal there. Now what I want to come across here. This year exam, PD 2017, I think I might have the question.
What is the meaning of leukemia?
Leukemia is a blood cancer. Like I have said yesterday, bone marrow cancer. Right? Bone marrow cancer, that's what. CSP, this is maybe 2017 exam. Yeah, this is 2017 exam. Yeah, this is 2017 exam. Right. Now look at here. Okay, question number 14. I'll just do this. Question number 14. Question 14. In the context of solving pollution, problems, what are the advantages of bioremediation technique? Right, this question was asked. Number one, it is a technique for cleaning up pollution by enhancing the same biodegradation process that occurs in nature. Number two, any contaminant with heavy metals such as cadmium and lead can be readily and completely treated by bioremediation using microorganisms. Right, number third, genetic engineering can be used to create microorganisms specifically designed for bioremediation. This was asked in the exam. It is linked with biotechnology, bioremediation process. Bioremediation is simply remediating or getting rid of or cleaning up waste or contaminants or pollutants from any site, maybe water or soil, with the help of microorganism or with the help of biological agent. That's what called bioremediation. I'll give you written part. That's what you have to understand. Now, statement number one, is it correct or wrong? Is correct? Why it is happening? Because in nature, in the soil, there are many type of bacteria, and there are certain type of bacteria like Pseudomonas, right, Clostridium. They keep on doing this kind of action there. Natural breakdown takes place. So first is right. What was second here? Cadmium, lead. What said here is that any contaminant with heavy metals such as cadmium and lead can be readily and completely treated by bioremediation using microorganism.
Larger in size than the microorganism.
Cadmium and lead larger in size than microorganism itself. So what microorganism can treat them into heavy metal? There's something called as here, I'll tell you one very important one, biomining. In biomining, what is it? Such kind of the bacteria which can be used for absorbing the metals, elements, right? And then for purification purpose, that is done there. So the same context, so what can be done there? See, remember that heavy metals mostly are taken. See here, does it use any word like here is what is contained heavy metal readily? Microorganisms given there. But in the nature, bioremediation can be also assessed through plants, trees and plants, which takes what's called phytoremediation. What is a very important word? Phytoremediation. Trees and plants' roots can take heavy metal like lead, cadmium, arsenic. That can be done. Right? The bacteria, that is the wrong statement. Right? Now see that's what I'm saying, that UPSC exam becomes difficult in the sense because you have only just 2 hours. And the moment you have to think and apply things, whether correct or wrong, and question is again takes time to read. You have lengthy statement given. Now coming to third one, genetic engineering. Correct. Today we can genetically engineer a microbe, a bacteria, and can go for such kind of cleanup. Normally remember that bioremediation is used for cleanup of oil spill and those kind of things. Right. Now see here why the question came, because it was in the news. Oil japper is there. And something more has been developed. Oil japper, one thing is that when you look at there, oil japper. What is oil japper? It is a culture of bacteria. It is a culture of 4 strains of bacteria which can break down, right, hydrocarbons. Right? What is the oil japper? It is a culture of bacteria. Let me write here. It is a culture of bacteria which can break down hydrocarbons. So it can be used for cleaning of oil spill, at the same time oily sludge. So right there. Is a culture of bacteria, and one of the preferred culture of this bacteria which comes into application is the Pseudomonas, right? So what is it? It is capable to break down hydrocarbons. Hydrocarbons can be broken down there, right? And this Department of Biotechnology funded research. So what it can break down? Hydrocarbons. So not only oil spill, treatment of oil spill, but also it can be used in case of oily sludge. Where do you find oily sludge from? Sludge means malwa, the west, where it forms refineries, oil extraction, they form sludge. Right, those kind of thick sludge can also be broken down. by using the such kind of bioremediation method, oil japper. Right, but again there was something in the news last year. There was again one strain of bacteria developed. Right, so instead of asking that name, question was asked with the basic concept. Right, so if you look at here, let's say, so if I just go into that, oleo, what is oleo? Oleo means oil, which contains 6 strain there. Remediation, oil space. New Delhi and was given there, deep water there. This way. Olivo, this name was there in the news. This was the last year in the news. Right, that's what this question came in the exam in terms of basic thing. That is a TERI, the Energy Research Institute, Delhi. Now this has what is it? Oilivorous S. Oilivorous S. That was I was trying to spell it out, but I was not getting that. Now what is given? Innovative by TERI. So I'll just zoom out there. So TERI has developed this particular method there. Not getting there. Now what is given here? A grand kind of movie kind of thing there. Oilivorous there. S. That is what you find here. And you can see here all this land and the areas they have used this particular thing. This was in the news. So instead of asking what was in the news, the question came. So that's what I said. What you are reading, the sources are there. But then you have to go to periphery. Because UPSC may not ask the name or directly oil japper, which everybody knows, but something from the basic biotechnology or any kind of area of science and technology question can be there. Okay? Now you can write about bioremediation, just one paragraph. I'll come to that part. Then after this, I'll come to gene therapy. I'll be coming to the method of gene therapy also there. I'll explain and then put into that part. TERI stands for the Energy Research Institute. Earlier it was called Tata Energy Research Institute, but then Tata has been Tata. Only the Energy Research Institute is called. It's Delhi-based organization. The TERI is there. Now you can write this. Bioremediation is the branch of biotechnology. Bioremediation is the branch of biotechnology which deals with the methods. Bioremediation is the branch of biotechnology which deals with the methods of solving the environmental problems. Methods of solving the environmental problems. Now you give a full stop there. It plays important role in cleaning. It plays an important role in cleaning the environment from pollutants. Cleaning the environment from pollutants by using the microorganisms. Such as bacteria and fungi. Cleaning the environment by using microorganisms such as bacteria and fungi. So even fungi is also used, right? Bacteria and fungi, give a full stop there. Bacteria, same paragraph. Bacteria are the most important microbes. In this process. Because they break down organic matter and nutrients. Because they break down organic matters and nutrients. I'll give you a full stop there. The next paragraph we write. Very important point I'm writing there. That's what the question came. Now to read. Break down organic matter and nutrients. Normally, what are bacteria? Decomposers. They break down the organic matter there. The next paragraph, now I'm coming to that. It is not necessary, very important point I'm giving. It is not necessary that all types of contaminants, it is not necessary that all types of contaminants can be destroyed with bioremediation. Right? Give a dash there. Heavy metals like can be destroyed with bioremediation. Can be destroyed with bioremediation. Dash. Heavy metals like lead and cadmium. Heavy metals like the lead and cadmium are not the type of contaminants. Which can be broken down. Are not the type of contaminants which can be broken down by the microorganisms. It can be broken down by the microorganisms. Right? By the microorganisms. Same thing here. This is I have detailed form. Here the box. Right, my book there. And there's question there. That's what I said that. The question I took is there because the same thing. I'll tell you that. This is where there in this chapter biotechnology, there is a box given. I've given the box bioremediation. And clearly mentioned here what I've given the statement form. Right? So sometimes in the textbook, it's not I'm saying that my book only. Any textbook, science textbook you read there may be something basic things will be given there. But again the question can be asked in the exam. Okay, ये बता रहा हूँ ये book का इसलिए क्या हुआ था कि this book which I prepared, though this book is very bulky, it's very thick, right? Because it consists of 550 pages. But I have covered everything which can be important for the exam. After going full stop, you write. It is the same paragraph, write this. A special type of contaminants, very important point I am giving. Special type of contaminants like chlorinated pesticides. Special type of contaminants like chlorinated pesticides can easily be broken down by bacteria. I'll repeat this. Special type of contaminants like chlorinated pesticides can easily be broken down by bacteria. Now taking first or last sentence. Similarly, oil spills can also be cleaned. Similarly, oil spills can also be cleaned by bacteria. Similarly, oil spills can also be cleaned by bacteria. Right? Cleaned by bacteria. Right? So most often what happens, wherever you learn, only the bacteria, oil spill, oil vapor that comes. Right? So this is what you have to remember for the examination because things are there. They may be coming to some other exams, may not be UPSC, but any other competitive exams, state pieces we are writing, and there's a bioremediation question comes. So that part comes into that. But now what I'm coming here, there's one area where question has not come directly in the exam, gene therapy. Bioremediation came this year, cloning came this year, but later gene therapy. So let me understand the things, then I'll come to the concept part of it. We have discussed stem cell therapy, now I'm coming to gene therapy. See, abnormal genes or let's say defective genes or genetic disorder, it is possible that through the use of gene therapy biotechnology method, we can correct those kind of faulty genes. Now what is the basic concept? Let me first give the key point, the basic concept, then I'll put in a paragraph there. What is done in gene therapy? I'll put in a flowchart that first time it was tried out in the US, gene therapy for a 4-year-old girl who was cured of what is called something called cystic fibrosis. A disease called cystic fibrosis. That was cured using this gene therapy, first time. When it happened in 90s, when in the US. There have been also attempts to treat cancer using gene therapy. Now what is here done is that gene therapy, what I said, it is where the faulty genes or let's say defective genes can be corrected. Now how the correction can be there? I will be telling you the mechanism part. So the one part is that we can repair that particular faulty gene. That can be part, or we can silence that gene. Right? There's something called single interference RNA technology, siRNA, which can silence that gene. If the gene is silenced, it will not be functional and replaced with a healthy copy. So what I am saying here, it can be done by different ways. Method one is that the repair. If we can repair the gene there, faulty gene there inside the human body, that can be one approach. I am saying that one approach is that. Or what we can do is that we can silence the gene expression. Not let that faulty gene to work, express there. And how to silence the faulty gene can be there? This can be done with the help of, there is something called, right, this is something called siRNA method. Single interference RNA, let me write in bracket there. Single interference, which can be used for silencing purpose. RNA is there, single strand which can help there. And what we can replace with the healthy gene. Let's say for example, in human body, because of that faulty gene, a particular protein is not being made. So that we can silence the gene and then get that gene into the chromosome, we can produce the protein. That can be another approach, right? Or something what comes here, that you have that totally repair or totally go with the new gene, the functional gene. Now what is very important, gene therapy is two types. I'll give you how it is done also, two types. The first type is called somatic cell gene therapy. You'll come across the word somatic cell gene therapy. Now what is somatic cell gene therapy? The normal body cells are corrected. What are the somatic cells? Normal body cells. That means the person can be cured of that particular disease. It's as far as the hemophilia, right, cystic fibrosis. The person can be cured of that. What is second aspect here? Germ cell. I'm using the word here, germ cell gene therapy. What is germ cell gene therapy? There, the reproductive cells, like egg cell or sperm cell, that can be corrected with the correct gene so that next generation offspring is cured of all the illnesses. And recently, right, one aspect of gene therapy has come through CRISPR. अभी ये न्यूज़ में रहा, राइट, न्यूज़ में भी रहा है अभी. इन यूएस में क्या किया, CRISPR यूज किया. क्या CRISPR क्या करेगा? यही करेगा, रिपेयर करेगा. Cut and the repair in this one. Where it was done? This one. Reproductive cells. So that the next generation offspring will be totally rid of, totally cure of the disease. That is what is now all making the news. Right? So what I said, two ways. Either you treat the person or you treat the germ cell so that the next generation is cure of disease. Two area comes there. Now how it can be possible? I am coming to how it can be done. How you can introduce a gene into the human body. So what is here? The thing is that is like this one. I'll put across here. The mechanism, I'm coming to the mechanism part. What is done is that certain type of viruses are used. Virus. What type of virus? Which infect human body, right? Human cells. And I'll give you one example, adenovirus. Right? So what is done is that healthy functional gene, let's say healthy functional gene, right? Let's say I am representing with this one, with this diagram. This is a healthy functional gene. That is packaged into DNA virus. You see, viruses are two types. I am talking DNA kind of viruses, which contain double strand, because gene is what? Double strand DNA. So let's say, for example, you take a virus, this one, and then the virus has got this DNA, and there you add this particular DNA. Let me just represent this one. I wanted to use another color, but let me make it bold. Now here is the human gene, right? And this virus, adenovirus, you just inject within the human body. It's injected there. So what will happen? This virus will infect the cell. Just like जैसे virus को attack करता है, affect करता है human body को, will infect that. And this gene, which is the human gene, will be transplanted there, will go within the cell. So what I want to say, virus act like a vector. But there will be problem. Why the problem will be there? You're using the viruses, and viruses are infectious. Their problem will be there. That's why gene therapy is a infancy stage. It's not a very advanced area, but today we are not using in a regular case. Selected cases have been tried out. Right now look at here.
No, only virus can't they use bacteria because
That can be, but viruses happens because why? What viruses do? Now, he has asked a very good question. Why viruses? Why not bacteria or some other kind of thing? Reason is that viruses, they enter the cell and they integrate the chromosome. In that case, the gene. Generally, virus कैसे multiply करता है? Viruses are like robbers. They're like robbers. They hijack the cellular machinery and they put their own kind of DNA or RNA and then multiply its copies, make its viral particles. That is the unique process of the viruses. Bacteria doesn't do that. That is the thing where viruses are used. But what I said, there will be problem areas here. So what can be next approach? So there are two upcoming approach in gene therapy. One is that nanotechnology. What I want to say, this is the one which has been tried out. But we can use nano biotechnology to go for some kind of a gene therapy. Now what we'll do in nanotechnology, let me put across there. How we can use that nano capsule. Now think that we have a nano material. Instead of taking virus, imagine that instead of taking virus, in the lab we make such kind of nano capsule. Right? And within this we put the human gene. There is a human gene which we have to insert. This human gene has to be inserted. And what we do this capsule, we put something on that, what I have said here, antibody. We can make this kind of thing. So that what will happen when the moment you put this nano capsule inside, they will go and bind to the cell and then deliver this gene inside the cell. That can be one another approach. And what I have said next approach there is CRISPR. CRISPR is also coming up there. Why not, like in China, is going to happen there. A team of scientists in China is going to treat cancer using CRISPR technology. That is also a way out. Right? Is it clear? So first I have given you the overview there. What exactly can be done now.
एक सेल को ट्रीट कर लोगे जीन से
हाँ.
तो जैसे कैंसर की बात है मल्टीपल सेल्स हैं जो डैमेज है एक एक सेल को ऐसे ट्रीट कर
हाँ तो वही है. दैट्स व्हाट वो डिफिकल्टी होता है पर्टिकुलर द सोमैटिक सेल. क्योंकि जो कैंसर है तो यू हैव टू ट्रीट ऑल द कैंसर. क्योंकि एक से दूसरा कैंसर फिर वो डिवाइड हो जाएगा. दैट्स व्हाई इट बिकम्स वेरी चैलेंजिंग बिकॉज यू हैव टू क्योर इट टोटली. देन इट इज पॉसिबल देयर. मैंने ये बताया मैकेनिज्म ये है. मैं फिर समझाया तो देन आई गिव यू रिटन पार्ट. व्हाट वी कैन डू, लेट्स से डिफेक्टिव जीन है एंड दैट कॉपी ऑफ द फंक्शनल जीन ऑफ द डिफेक्टिव जीन एंड वी कैन डू इन द लैब, वी कैन पैकेज इन अ virus, infective virus. That is there is a viral DNA, and we have added with the viral DNA human DNA. Right, we have added there, and then put it back to the virus. So virus, when it goes to the human body, जब वो body के अंदर जाएगा, infect the cell, so it will integrate this DNA, right? And there, this human DNA will also get integrated, right, in the chromosome. Yes, the human DNA will integrate it there, right? I'll put it there. You can write there. Just like molecular scissor.
तो एक एंजाइम को
ना मैं भी enzyme नहीं प्रोड्यूस करता है. CRISPR is basically consists of two things. One is the sequence, and second is what called an enzyme. Enzyme helps in cutting. And what the sequence use? Recognition. It recognizes the site where the strand has to be cut. So this is something called CRISPR technology. Basically, what? One is the RNA. There is an RNA which basically identifies the which part of DNA has to be cut. And then the enzyme comes, bind that part and cut it like a scissor. That is called Cas9. CS9 जो है, is called CRISPR associated protein or enzyme, which cut the part of DNA. That part comes, right? So CRISPR, that is what comes into that. But actually, main aspect is that the place where you want to make a nick or cut, that has to be recognized. So that's where CRISPR comes into that. Gene editing tool that comes there. Now you can write the gene therapy. I'll put across gene therapy there. Now write this. I'll first give this. Gene therapy is a technique. Gene therapy is a technique for correcting defective genes. Gene therapy is a technique for correcting defective genes. Defective genes responsible for disease development. Responsible for disease development. Responsible for disease development. Now taking full stop there. Responsible for disease development. Now you write this. Researchers may use one of several approaches for correcting faulty genes. Researchers may use one of several approaches for correcting faulty genes. Give a dash, hyphen, number one. A normal gene, dash, number one. A normal gene may be inserted. Normal gene may be inserted into a non-specific location. Into a non-specific location within the genome. Non-specific location within the genome, genome, to replace a non-functional and non-functional gene. Non-functional gene. Let me explain also there. I'll give you all the but give explanation. Non-functional gene. Give a full stop. Let me explain. What is that? You insert this kind of gene which will go to any other chromosome or any other location on the chromosome. Not necessarily that the part where the faulty gene is there. And then will provide the particular kind of function there. This is the most common use. What I've given this is the most commonly used. So you can write in that sentence, that part, give a full stop there. You can write, this approach is most common. This approach is most common. So I've clearly given the word non-specific location, anywhere. In the chromosome it can go and can have the function. Right? So this is the most common approach. Now to give full stop, second one, number two. Now write number two. An abnormal gene, number two, an abnormal gene could be swapped, SWAPPED, could be swapped for a normal gene, could be swapped, SWAPPED, for a normal gene through I'm using one technical word here, homologous, HOMOLOGOUS, homologous recombination. Homologous recombination. Homologous recombination. Now let me explain what is this homologous recombination. When cell divide, right? When the cell divide, so what happens? Chromosome also exchange material. So what can happen in the process of exchange of material, homologous recombination? Right? There will be what is called crossing over, swapping. So let's say when the process of cell division happens and crossing over happens, abnormal genes get swapped, replaced by the normal one. And then we can block the abnormal gene. That part is it. Again, that is all method tried out. That's what I said, gene therapy is today in infancy stage. Today is not very commonly used. This is what the scientists have done that. ये सब इसीलिए पॉइंट मैं आपको दे दे रहा हूँ क्योंकि in case UPSC exam, if such concept is given statement form, so at least you know that this can be a way out, which can be done there, right? Now third one, number third you write, the abnormal gene could be repaired. The abnormal gene could be repaired through selective reverse mutation. The abnormal gene could be repaired through selective reverse mutation, comma, give a comma, selective reverse mutation, comma, which returns the gene to its normal function. Which returns the gene to its normal function. Which returns the gene to its normal function. Now let me explain there. First of all, you should understand what is mutation. What is mutation? Sudden change in the DNA. Again, remember that any organism, including human beings, always the repair mechanism works. But during the DNA multiplication repair, always their fault happens. What is fault? Called mutation. Changes happen there. So what I've given the approach there, what I've given the abnormal gene could be repaired through selective reverse combination. You identify where that abnormal gene is there and you go for replication, ulta, opposite one. And there through the mutation, you change the list. For example, my diagram bana do. Let's say ऐसा मानो आप, just an example I am giving that. This is here A, just for example, T, G, just only for expression purpose. तो complementary होता है तो T, A, C, ठीक है? क्या इसमें गड़बड़ ये है? Let's say these two are wrong. Let's understand. You have a DNA sequence, there's a gene there, but this information is wrong, that's why the gene is wrong. Gene is not working. So what we want? We want to change it, remove it. ठीक है, इसको remove करना है. तो अब क्या होगा जब DNA multiply करता है? Let me just so when what happens, the strands they separate. ऐसा होता है. Let's say I'm just giving example. So A, T, and T, G, right? And here you have T, A, C. तो जब ये multiply होगा, when the another strand will be made, there you introduce the mutation. ठीक है, वहां पर आप क्या करो म्यूटेशन कर दो. क्या मतलब हमें ये रिप्लेस करना है with the let's say CC or what is correct one, correct sequence, and that is brought about. This I am giving too much of detail, just only to give you understanding that if reverse mutation. अभी जो न्यूज में आया हुआ है CRISPR they are repairing. So what they are doing, they are cutting that part so that when the enzyme works, they repair it, they fill the gap with the correct sequence. ये भी CRISPR में किया जा रहा है. This approach has been being tried out, right? And the last one, the fourth one you be right. That can have side effect also. Right? Can be. Now fourth one, fourth one, right? The regulation of a particular gene could be altered. What is the fourth approach? The regulation of a particular gene could be altered. ALTERED. By making gene turned on or off. By making the gene turned on or off. That means expression. If you can regulate that change as well. Okay? Then what I said, what is the mechanism? Using such viruses. I have given you 4 approach, but again remember that I have not added CRISPR, but remember that that's the way making the things can be done, but it is normal ways that I've added. Now you can write the last paragraph, next paragraph write. Last paragraph write there. In most gene therapy studies, like how it is done there. Next paragraph. In most gene therapy studies, a normal gene is inserted. A normal gene is inserted into the genome to replace an abnormal, inserted into the genome to replace an abnormal disease causing gene, with a vector, VECTOR, with a vector such as DNA containing viruses, for example adenovirus, to deliver the therapeutic gene to the targeted cells. Deliver therapeutic gene to the targeted cells. This is what the commonly used there, right? And now same right in paragraphs. So let me give you complete information because this area has not been asked. So in case question comes, besides, right, last same paragraph. Besides virus mediated gene delivery systems, besides virus mediated gene delivery systems. I'll repeat that. Besides virus mediated, MEDIATED, mediated gene delivery systems, comma, there are several non-viral options. There are several non-viral options for gene delivery. For example, use of nanocapsules. For example, use of nanocapsule with the healthy nanocapsule with the healthy functional gene, as delivery molecule. Let's give a full stop. Okay. So there are disadvantages also, but actually this is a general concept which I've come across. Right. So we have discussed that part. Now I'm coming to, well, I'll show you one video also there. Yes. Yes. That is wrong. That वो गलत है. That is you're right there. We can create mutation and make our children more intelligent. Because what we are doing the selective one. Selectively we are doing that one. So that is the part what. See what is it? We have all the machinery. Through mutation we try to make that kind of gene which can give that kind of high intelligence to the person there. So we are changing genetic sequence or creating that gene which can add that kind of function there. वो उस तरह से किया जा रहा है. लेकिन वहाँ सवाल यही एथिकल सवाल उठता है. क्या ऐसा करना ठीक है कि ह्यूमंस को वी आर मॉडिफाइंग, trying to become very selective in human beings? That is really like if you ask any ethicist, they'll say that man is trying to become like God. You are trying to develop what we want for our own benefit there. So that's where the challenge lies. Right? तो ये सारी चीजें हैं. CRISPR has opened an entire Pandora box. सारा डिबेट ओपन कर दिया वो. Right? इसलिए you see that question में हमेशा 2-3 I have found. You will just look at last 6 year paper. 2 or 3 questions are always linked with biology and biotechnology part. Particularly applied biology and biotechnology. That is what asked in the exam. But I'll show you the gene therapy video. That's what I want to come across there. Let's see the gene therapy video there. Whatever I'm covering is the more than sufficient. I'm giving you everything on that. Right, so don't worry about this. Whatever you are learning here, that is just keep that point, and this you just revise before the exam. But we can do the tissue culture and then take to the field. And bamboo is a poor man's resource because of furniture, house, everything is done there. But what is the thing here? The gestation period is very long. Right? Flowering happens very long. But we can go for tissue culture. So the number of type of crops and plants are being now cultivated using such kind of centers have been there. Thousands of plants are there. Right? Let me show you the photograph. There is a photograph there. This is Gwal Pahadi one, Department of Biotechnology. You see the images there. DBT, Gwal Pahadi, tissue culture center. Micropropagation technology lab. Technology part Terry is there. DBT and this was supporting upcoming. Tissue culture facility is there. It has come in the NIC. It's given there. They have developed this system and their certification also there. And they're not one or two, hundreds of plants are being tissue cultured in such kind of case, right? So now you can write there. I'll come to that. I'm coming to this. It is a technique. Just write one paragraph. It is a technique. It is a technique of growing tissues or cells of. It is a technique of growing tissues or cells of multicellular organisms. Cells of multicellular organisms in an artificial environment. In artificial environments. Now, after giving full stop, artificial environment, I'll write. In plant tissue culture, comma, the medium is controlled. The medium is controlled with required amounts of nutrients. Is controlled with required amounts of nutrients and phytohormones, PHYTO, required amounts of nutrients and phytohormones, PHYTO, hormones, HORMONES. Now begin first of. In this technique, comma, meristematic, MERISMATIC, meristematic tissues from a plant is grown into callus, 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 grown into callus, right in bracket there, right, undifferentiated mass of cells, bracket closed, give a full stop there. Later the callus differentiates to give rise to the plant, plantlet, PLANTLET, small plant, which is a clone of the original plant, that is similar to that plant, which is a clone of the plant there, of the original plant. Now give a full stop there. Last sentence there is it. It is used regularly for micropropagation of crops of hybrid variety, give a full stop there. Now after that you give rise, just write there. It has crops of high yield varieties, which I've said, give a full stop there. After that, it has vast potential in dash, give a dash, number 1, give first, vast potential in dash, number 1, clonal propagation in a mass scale, spatially, clonal propagation in mass scale, SCLE, spatially ornamental and horticulture plant, Second point, important point, number 2, obtaining disease-free plant material. Number 3, producing biologically active, number third, producing biologically active compounds, producing biologically active compounds for pharmaceutical industry, producing biologically active compounds for pharmaceutical industry. Right? Another very important point, fourth one, genetic engineering, in which foreign genetic material, genetic engineering where foreign genetic materials are introduced with the help of culture medium. I'll repeat that. Genetic engineering where foreign genetic materials are introduced in the culture medium. So let's say the culture can act in terms of transfer of such genetic material, the culture medium. Now, fifth point, producing crops which can withstand high salinity, aridity, high salinity, aridity, 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 and other extreme conditions. Aridity and other extreme conditions. Right? And the last one, 6 point, number 6, conservation of endangered plant species. Conservation of endangered plant species and preserving by cryopreservation. Preserving by cryopreservation. Now what is cryopreservation? Just write in bracket cryopreservation. Freeze, F-R-E-E-Z-E, freeze dry method. Freeze dry, D-R-Y, dry method. Freeze dry method. That means cryo means very low temperature. What we can do? We can make this kind of plant and then preserve it at very, very low temperature. The cryopreservation comes, right? Few things now I'll just put on the whiteboard. There I'll not dictate the notes. They'll just come into that part. I have said in the classroom, there is one branch of biotechnology called bioinformatics. Right? Bioinformatics part. Now in the bioinformatics part, and I've said in the classroom that India has huge potential for this field, the bioinformatics part. Now I'll put everything on the board. This is what you can write. The first thing is that is use of ICT, use of computers and information technology to manage biological data. So you can say that this is also one of the part which is linked with big data analysis, because biological data or biotechnical data can be very huge, can be very large. Let's say for the human genome project, human genome. The human genome, if you just take example of simple human being, so it consists of chromosomes and we take nucleotide base. So 3.2, 10 to power 9, right, that is the nitrogenous bases there. ATG and all this, we have this kind of bases there, a single cell. Imagine we have got trillions of cells and we have to store all the information. Let's say for example, it will take a number of directories, font directories to write all this information. But today computers like supercomputers can store that data. We can analyze the data, like by writing a program, software program for similarity, dissimilarity, all this is possible. So what is the bioinformatics? Where first is the storage of data in computers and then through software analyzing the data. That comes here. Now what is second aspect comes here? Bioinformatics research, right, has gone into different areas. Now what are the different areas in this case? What I'm writing first is called proteomics. What do you mean by proteomics? Let's say this term comes in the exam. Nowadays the word ohm comes, O-M-I-C-S, omics. Omics means study, right? And when you say proteomics is nothing but study of structure and function of peptide and protein. Study of structure and function of peptides or proteins. Let's say a scientist finds new type of protein from any of the organism and wants to learn the structure of the protein and the function of the protein. It will take a lot of time. In the lab, it will take a lot of time. A biotechnology has to spend a lot of time. But today it is possible through bioinformatics. Why? Once you sequence the protein, what is protein made up of? Peptide. What is peptide made of? Amino acids. If we have a sequencing one, just like Google, what we do in Google, we write the word. And what does Google does? Search entire database. And whatever the hit comes, similarity, the information comes in. So there's something called protein data bank, PDB. I'm just giving you that how bioinformatics works. Like here, if you take example of PDB, European countries have a protein data bank. Let's say a biotechnology find a protein, do the sequence of protein, just put that information PDB. PDB will search entire database, whatever known proteins are there, and give you the result that the protein which scientists have found out is similar to which type of protein. So that gives new direction to research. That is what the PDB comes. So this is protein data bank. And they have got this kind of software functions where one can do the search analysis part of this. You will come across in newspaper that scientists keep on doing DNA sequencing and genome projects. So human genome being one, but there are other genome projects of plants and animals being carried out. Why scientists are doing such kind of genome project? To understand the characteristics of that particular species of plant or animal, so that a new variety can be developed through genetic engineering or something, crossbreeding, interbreeding can be developed. So what I'm coming second area is called genomics. What comes here? Genomics. So just as I said, whenever the word omics comes, collection or study of that. So what here means? Here means study of structure and function of gene. Structure and function of genes. So that's kind of branches will come across. Right? These are two very emerging areas of biotechnology, bioinformatics part. Now what I'm coming here, if you look at application of this one, and particularly as I said, such data can be done only, so what is done here? Use of supercomputers. That is done through use of supercomputers and advanced computing there. That can be done in the research part. Supercomputer, advanced computing. ठीक है, मैं आपको कुछ एग्जांपल इंडियन कॉन्टेक्स्ट पे भी देता हूँ, and I'll give you those current example also there. Which agency or which body in India built supercomputers? CDAC, Pune. We have supercomputing facility for bioinformatics research in India. I'll give you one example. If you look at Indian aspect there, CDAC has got something called here, which is called, in terms of their what called is a bio, right? Bio, this bioinformatics one, I think Param, not from there. It is a center in Pune only. They have given the name also there. This Bio Param or something has been given. Let me just give you there. This name which has been there, which is a specialized center for only bioinformatics research, not for structural design or aeronautical engineering or defense or something like that, but only for this one, as comes only for the part of this bioinformatics research there. CDAC, Pune. This part is there. This, because you know this Param always comes into that part. Specialized center is there. High performance computing you'll find this. Bioinformatics this part. This font is small, but I'll just read out there. Bioinformatics resources and application facility at CDAC is an effort for providing high-end supercomputing facility to the researchers working the areas of bioinformatics. This is what is given there. So we are also doing in the country. Use such supercomputers. Right? This is what is given, BRF, which is there. So what is done in this case? Now I'm coming to the application part of it. Why? What is the application? we can write software, we can simulate, do the modeling, and make that kind of molecule what we are looking for. And then we go for lab testing, lab working there. So I'm coming to application part. Bioinformatics have the potential applications. The first area what called drug discovery program. In our country, we are carrying out this bioinformatics research for finding a novel drug for cancer or any kind of deadly disease there. Drug discovery. So what we can do? We can simulate that particular drug molecule, and then we'll go for making in the lab, chemical synthesis. Second application comes, I'm writing very important word, phytogenetics. Now what is phytogenetics? Evolutionary biology. I'm writing the bracket here, evolutionary biology. I'll explain with an example. If we want to compare how human is similar to a mouse or chimpanzee or ape, if we are trying to do in the lab, it will take many, many years. Phylogenetics, I'm sorry, sir. Phylogenetics, not phyto. Phyto means plant. Phylogenetics. And what can be done? We can make a tree of life, right from bacteria till humans, like this kind of structure you'll find. Starting from bacteria, how the other species have evolved, and where the man comes in, similarity and dissimilarity. Today it is possible in the computer. We're using the information technology, we can use this kind of analysis. Let's say we take human and we take 10 organisms, and now we want to find similarity and dissimilarity. Very surprising is that we are almost 80 to 90% similar to a mouse. But a mouse is very small, but we are similar to that. At what? DNA level. That kind of analysis is there. Structural level is not there. Right? Chuha or Manav, structural is not, but genetic level we are same. That's why many of the experiments are done on the mouse, chimpanzee, where the similarity aspect comes. Now what I'm coming, the third year. Third very important aspect which you have to remember that, pedigree analysis, pedigree. What is pedigree? One is add is a dog food, right? But here's something else. What is pedigree? Family history. Like for example, hemophilia is common in royal family of England. So how that royal family hemophilia has come? 4 generations back or 5 generations back, 10 generations back. That what. And you know that when you solve reasoning question, so how the reasoning question you have this? A male marries a female, right? And then has children there, daughters and sons and everything, right? This is how the family tree is built. This get married to another one there, and they have the other children there. Who's the uncle of whose? So how the gene has passed, right? So that can be done there. Genetic disorder part of that. That part is analyzed there, right? Apart from this one, what I've said is that we can go for this kind of proteomics and genomic studies, database and all this, right? So bioinformatics, since we have enough of manpower, so we can go for this kind of analysis there, right? Now what I've said here, we are doing for drug discovery program. It has come in Science Reporter and the place. So I'm just opening that particular website. There's something called OSDD. CSIR has got something, open source drug discovery program. CSIR, it has come 2 or 3 times in Science Reporter. OSDD, open source drug discovery program. And what is the word? Open source, that is free. Right, this database is developed. So any scientist in India or outside can come together and they can work for tuberculosis, HIV, malaria, and can develop the drug. And they're collaborating through using bioinformatics also there. So let's say if question comes in the UPSC exam, this particular from the government aspect. So please be aware of this particular open source drug discovery program. Now see clearly here, what is given? SysBorg. Right, what is SysBorg? SysBorg is a OSDD cyber infrastructure for collaborative research. SysBorg has over 7,900 participants from 130 countries across the world. And what is said, join SysBorg in order to know more about the ongoing activities of OSDD. Now OSDD chemistry. Now what is this? Is the web interface for large-scale synthesis of diverse chemical compounds to screen them against the TB and malaria. Very clearly given. For tuberculosis, malaria, to find out new type of drugs. There's a collaboration platform is there, right? OSDD Chem. And what is further given? Log into and this part is given. Now current event and updates you have. So since it has come in Science Reporter, but not in the Hindu paper, will not come across occasionally. So from that thing, I have linked with that. One is that Param in terms of supercomputing facility we are using for bioproduct research, and second is this particular OSDD program, which is a collaborative program there. Clear? So this is how you have to link with biotechnology part. Whatever I have covered, genetic engineering or cloning or gene therapy, stem cells, and all this part. These are the where it comes in the newspaper and all this. So at least you will be comfortable when you read those kind of areas. You will be more comfortable for part of that. ठीक है यहाँ तक clear है? Is it clear there? Now let me come to the last part, just half an hour there. We will be going to the part of health aspect linked with the biology and health aspect. Now, how do you define disease? It will come to basic things. Then I'll come to the examples and everything there. So disease, how disease? How many types of disease are there? Communicable and non-communicable. Example of communicable diseases? Common cold, okay. Apart from common cold, HIV, okay. But you have to classify, categorize. You have to categorize this. How would you categorize the entire? So I'm now making a flow chart here. See, when you talk about diseases, we can learn into the following ways. The first one comes here, infectious or communicable diseases. Now one way is to learn is that those diseases which spread through food, water, air, right, those kind of, or right. So let's say here food, waterborne or airborne. Give me example there. Let's say cholera, which is a bacterial, right? Typhoid, again a bacterial. Air form, TB, tuberculosis. Example I am giving that. Second way is what? It happen through contact. Right, contagious there. Now give me example there. Any example? One good example, leprosy, which is a bacterial, mycobacterial leprae. Right? Contact can be flu viruses also there. Very well. But this is one example I am giving from leprosy point of view. What is next comes here? Next what comes? Vector borne. It can only happen through animals or insects. Right? It will not happen through any kind of contact or food or water, will not happen. So vector borne. So when we say vector borne, so there malaria comes. Example, let's say malaria, then now this is dengue. All these are making, now see here, encephalitis. Now a lot of news is coming. And now the report has also come that oxygen is not the cause. Lack of oxygen is not the cause. Because children were already suffering. And again the problem with what doctors are still in dark. Right, so scrub, but now what have come? Scrub typhus. This is what is made in the news. It is not encephalitis but scrub typhus. That is type of disease which is causing the death of the children in Gorakhpur. That is what is making that. So when you read newspaper about any kind of this one, chikungunya or dengue or malaria, encephalitis, you have to know from that what perspective, what type of causative agent, how it spreads. Right. Now let's say here, another way in communicable disease what? Through pathogens. What comes the next here? Pathogens. And their classification, bacterial, right, viral, then what comes? Protozoan, and then you have fungal disease, right, which can cause through bacteria, which can cause through viruses, which can be caused through protozoa, or which can be caused through fungi or fungus, right. This one comes. Now there are diseases which do not, or which are not contagious, or which are not infectious. They are called NCD, non-communicable disease. Now non-communicable diseases, now classification here, first comes here, deficiency disorders. What I am using the word here, deficiency. Because this is not contagious, so this can spread through deficiency of certain thing there. And we can divide into first A, hormonal deficiency. Give me an example, diabetes, like lack of insulin. Right, that part comes there. Then, if you look at another, vitamin deficiency, vitamins, example are many, like night blindness, vitamin A, right? Then scurvy caused by vitamin C. Then arthritis, right? Vitamin D. So rickets, vitamin D. All these are there, vitamin deficiency. And third comes what? Minerals. Let's say iron causes anemia, right? Then iodine causes goiter. So example here, let's say here, anemia. See, the cause of anemia could be many. One factor could be lack of iron in the diet. But anemia can be sickle cell anemia also, with genetic disorder. Anemia can be because of vitamin also there. Right, B2, B12, cobalamin. Cobalamin, what is it? Cobalamin. The B12. Well, pernicious anemia, pernicious. So there are different factors. But here what I am writing from iron deficiency. Then goiter, iodine. Apart from this, what comes here? Another metabolic disorder, metabolism. So what is basically linked with the metabolic disorder is in terms of what people suffer with obesity, or sometimes for not having become obese, lean and thin. Again, that comes the metabolic disorder, right? So one good example which is making here, that is the obesity, and obesity leads to hypertension, right? Cardiovascular diseases. All these are factors there. What comes the next one here? Cardiovascular diseases. Heart attack. What is called in technical word? What is it called there? Myocardium. Huh?
Myocardial infarction.
Infarction, right? Infarction. INFACTION. Infarction. But there is something also there. Arteriosclerosis. What is arteriosclerosis? Thickening of walls of arteries because of deposition of cholesterol. Right? Calcium there or cholesterol? Both will be the cholesterol is salt part. But again, the cholesterol comes into that. Right? So these all words are there. So these again will not spread. If you have cardiovascular disease, it will not spread to another one. Right? That happens to a person. Then very, very important. I am writing here. I'll put on the top there. Cancer. Right? Though cancer can be caused by viruses, what is called oncovirus, like hepatitis can cause cancer. But normally cancer is an individual basis which happens there, tumor and cancer. And then what I'm coming here, mental illnesses. Mental illnesses also. So mental form of disorder, mental illnesses, the people take it very lightly, depression. These are like depression can be mild and it can be also severe. Severely dangerous. Right? और आजकल बहुत कॉमन है बॉयफ्रेंड गर्लफ्रेंड husband wife right boss and all this in the office boss will be there so all this is happening there their IAS officers they are becoming depressed and there is our case study IAS committing suicide right why depression here people don't know when they become victim of depression slow onset. And what I'm writing here, another thing which students suffer, chronic anxiety, but we take it very lightly. Tension करना चलो अच्छी बात है, लेकिन कब वो anxiety can become chronic anxiety, nobody knows. तो क्या होता है? 2016 prelims exam not clear. 2017 prelims exam not clear. No, prelims are clear, mains, right? Interview no call. Year after year, right? What happens there? Anxiety. This exam brings the anxiety, and the chronic anxiety leads to again the depression thing there, right? So there we take it very lightly. We say, oh, koi baat nahi hai. But it's an illness. But when there is an illness there, it needs a treatment. Gangaram Hospital, which is very nearby, right? And you don't know, there are cases of psychiatric treatment also there. People go for that kind of thing, right? It happens there. Right? There's one friend of mine who goes for counseling there and says that कि बच्चे अपने माँ पिताजी के साथ आते हैं, right? So that kind of things can happen to individual, but I'm not talking UPSC exam. It can happen in general also there. Anxiety. Now what I'm coming, the dangerous one, which you know that, Alzheimer. What happens Alzheimer there? Alzheimer, there what happened, brain cells, brain tissue degenerate. It is also called degenerative diseases. Brain tissues, they degenerate. जैसे हिंदी में कहते हैं सठिया जाना. Right, when you cross the age of 60, so you have the symptom like Alzheimer. What is symptom like there? सठिया जाना. खाना खाने के बाद पता ही नहीं खाना खाया है. Right, the person there asks for another. हाँ वो भी हो सकता है जब हाँ दम मारू दम. That is what happens. Alcoholism. People go into the what is the hallucination mode. That goes. But here I am talking in general. What happens there? You forget what I'm saying there. When you are in a high state, you do not forget. You remember things. Right? You remember more than the normal state. That's what happens. But there in Alzheimer case, people forget. They lose the memory. Memory loss is there. Right? That's what said. What happened? The people age. Aging factor. They don't know even they have taken the food. Where they are standing. They forget that. So what happens Alzheimer? There's a brain tissue loss. And that causes dementia. Memory loss. And slowly everything. Because this is the master point for entire body there. And what happens Western world, where is more common? Western. Indian society has but not that common. But Western world you will have common. What they do there? When the person is diagnosed with Alzheimer, they invite the near and dear ones. Throw a party. Because the person will very soon will go into exclusion. Will forget everything. And then finally died. That is the pathology disease there. A lot of research is going on. Genetic cause of Alzheimer, who can get their family history, a lot of studies are there. But still, there is no treatment. You cannot have any treatment for Alzheimer. Then you know, schizophrenia. What he was saying that what happens schizophrenia case there. Hallucination, delusion, right? A daydreamer, this is good. So what is the schizophrenia? It can be mild and severe both. Mild case it will be very difficult to identify. Right? कभी अगर आपके आसपास कुछ आवाज आए या आप किसी से बातचीत करते हो, ठीक है, किसी पूछ लेना कि वो सही में आदमी है कि नहीं है. Right? One very good example is John Nash. I am very sure you have watched that movie, A Beautiful Mind, played by Russell Crowe. The actor, gladiator actor, he played the role of Russell Crowe played the role of John Nash, who won Nobel Prize for game theory, right? Mathematician from Princeton and this. Harvard there. So here that is shown in the life of John Nash. What exactly happens to schizophrenia? The person thinks that he is being conspired. They act like detectives, like right, MDA agent and all this fun. That is what happens there. Hallucination, delusion. Why this kind of disorder happens in schizophrenia? Neurotransmitter. There is something called acetylcholine, neurotransmitter, dopamine, which they become abnormal. The people they develop this kind of symptoms there. Now I am coming to another one, bipolar disorder. There is another type of disorder, mental illness, bipolar disorder. What is bipolar disorder? It's like something like Dr. Jekyll and Mr. Hyde. So what happens? The person goes into low phase. Silent, everything, low phase. And then one day starts ringing up everyone, calling up everyone. Aaj raat party hai. And they invite everyone there. And they spend all the money, next day bankrupt. Right? Again, back to the square. There's bipolar disorder. And many of the serial killers have been diagnosed with this kind of disorder there. Right? So they have a high stage, then low stage. What is called bipolar disorder. Again, it happens because of what? What I said? Neurotransmitters. They are responsible for that. Correct? So mental illness can be a normal thing which we do not recognize. But can be the others which symptoms can be there. Now what comes the next one? Genetic disorder. Genetic disorder. Now genetic disorders can be both chromosomal and gene based. Chromosome abnormalities could be there. Where the chromosome is not normal. Let's say Down syndrome, Klinefelters, all this comes there. Right? तो यहाँ पे प्रॉब्लम क्या आता है कि यूपीएससी में जो क्वेश्चन आ रहा है वो सब करंट से ही पूछ रहे हैं डिजीज का. जैसे जीका वाज आस्क्ड, इबोला वाज आस्क्ड, स्वाइन फ्लू वाज आस्क्ड. Right? It's not asking you the normal biology and based on that. But again, still be prepared. right? तो वहाँ पे क्या है rectification, you require rectification, knowing the disease, cause of the name of the bacteria, name of the virus, all those factors there. So what I'll do in this case, I'm now going to very important website. You can write this anytime. Let's say remember that this is what I've given first overall on that view of that. But anytime you want to learn about a disease which you come across in newspaper, and I'll demonstrate with an example. Government of India, Ministry of Health and Family Welfare, has developed a very good database. Right, this is called National Health Portal, NHP. National Health Portal. This is what is there. And there you'll find A to Z of diseases. Right, when you open that, you'll find A to Z of diseases there. The link is given. Right, see here, Digital Initiative, National Health Portal, Ministry of Health and Family Welfare. And what is saying here? It's saying that objective, improve health literacy, improve access to health services, decrease burden of diseases through awareness. And then you have here National Health Portal, hospital near you, ambulance services, blood banks, emergency helpline, healthy living. Links have been given. Okay? Now let's say here, I am now coming to this A to Z of diseases. Yes. This is A to Z. Now if you just open this one, now let's say for example Japanese encephalitis. This is what is making the news there. And it is a multilingual database. You'll find in the part of the multilingual database, there's also given the search. Now what do you find in this case? When you open this one, right, let's say this part, it gives you the cause, symptoms, if there's any kind of prevention strategy, any kind of treatment, medicine, vaccines, all is given in one place. So let me just open this one. Very clear introduction, symptoms, causes, diagnosis, management, complications, prevention. A very simple language, any person can read and understand. Right? Let's say for example, here, let me zoom out so that people can read from the back also. Now say introduction part. JE is a viral disease that infects animals and humans. So what is the keyword? Animals and humans. Animals also there. Right? Last time the question Zika came. People were only knowing microcephaly GBS. We are not knowing that apart from Aedes aegypti, it can also be through sexual transmission. But it's given in the NHP there. Now look at here. It is transmitted by mosquitoes in humans, causing inflammation of the membranes around the brain. Brain fever. And very clearly given mosquitoes. Vector borne. It comes a vector borne there. Now what is here? Japanese encephalitis is a leading cause of viral encephalitis in Asia, generally spread from Western Pacific region in east to Pakistan in west, from Korea in north to Papua New Guinea in south. Asian is a more. Right? Africa or South and North America is an Asian kind of disease. So that is the location given area wise. That can be part of the question in the exam. Now let's say here, next one. Japanese encephalitis is a disease caused by flavivirus. Now, name of the virus will not be asked in the exam. Arbovirus, flavivirus, that is not that important. But regional area you should know that affects the membranes around the brain. General infection caused by Japanese virus are mild, fever and headache, or without apparent symptoms. But sometimes 1 in 200 infection can result in severe disease characterized by rapid onset of high-grade fever, headache, neck stiffness, disorientation, coma, seizures, spastic paralysis, and death. This is what happens there. Now, if you look at here, I'll give you the part. Let's say here, there's symptoms. So I'll not go to symptoms, causes here. Now look at here. Flavivirus is given here, and with both animals is now through an infected mosquito. Now, which mosquito caused that? Which mosquito? Culex, Aedes. Which comes in that? Aedes only. That's what it is. You have to know in that part. Now look at here, diagnosis. What is given? Blood test. To know the antibodies present in the blood. So which testing? ELISA. It's not written here, but ELISA will be used. Blood antibodies. What is said the second? Lumbar puncture. What happens? There's this one. They do the puncture. Cerebrospinal fluid, CSF. Those kind of fluid is taken to find out the virus. And this has been done in the case of Gorakhpur also there, to diagnose whether the fever is because of J, because still the doctors are in dark. Whether that children are dying because of this virus or something else is the infection. Right. Now is what is given? Scans in case of brain encephalitis. Computerized tomography, CT scan. Right, series of X-rays in them. MRI scan, this is what is given. Now comes that, can there be any kind of prevention there? Management part, there's the management part. Let me come to management part here. Where's the mouse? Mosquito control. Or understanding the health and all this. Right. But let's see the prevention there. This one there. It is given at individual level, vector density and all this. Reduce mosquito bites during peak biting hours. Same as what we do for dengue and chikungunya and all this. That area is given. In the community, right, malathion fogging during outbreaks. Same we do for mosquitoes, malaria or dengue or something. We spread those kind of things. Sensitizing and involving the community for detection of vector. This part is given. Prevention during travel, during pregnancy, some do's and don'ts. That's what is given. Right? So when you come across any such name, you just visit this NHP. I will have all the information about any kind of thing there. Like for example, malaria. So what is the causative agent of malaria? Plasmodium falciparum, Plasmodium vivax, Malarial ovary. 4 types are there. Right? Out of which Plasmodium falciparum and vivax are common in India. Malarial ovary you will find in Africa. There you will find more common there. But in India, we have falciparum there. What happens in the case of malarial parasite? What happens in that case? It goes into RBC. Right, the parasite multiplies, grows, like RBC there. And that is affected there. Right? So again those part, like for example, let's say there's a question which came in the exam, I'm just coming to that. Zika. See, Zika virus, right, and the Ebola virus. Zika is a vector-borne. Ebola virus, if you look at this Ebola virus, I've written vector-borne, but when you look at Ebola case there's something called zoonotic. Zoonotic disease. What is zoonotic disease? Which spread from animal to humans. What is zoonotic disease? Which spreads from animals to humans. And very good example is Ebola. Right? Only question in the exam was that Ebola spread was reported where in the news? Sierra Leone, Nigeria, those kind of names there. And you find that it has also directly been asked from this particular part there. Let's say if I just go into Ebola. How the name Ebola has come? You are aware of also why the name Ebola has happened? Zaire, Congo. There is Ebola forest. And it came from animals, bats and all this, to the humans there. Right? Now if you look at here, Ebola virus disease, EBD. This is what the image has been given. What is a method of transmission, secretion or other body fluids of an infected person? It is clearly mentioned there. Then what is given? Direct contact with infected blood. That can cause the Ebola. Then what is Ebola virus disease? Is one of the viral hemorrhagic fevers. Just like dengue, it causes hemorrhagic fever. And what is said, which affects a person's blood system. That concept you have to know, blood system. Then what is given? Symptoms. Fever, headache, weakness, joint and muscle pain, diarrhea, vomiting, pain in stomach, loss of appetite. An extreme case, it may lead to internal as well as external bleeding. That causes Ebola case. And highly infectious. It is highly infectious in that case. Facts about Ebola. Can be transmitted through water. Can't be transmitted. Can't. It will not be transmitted through water. Cannot be transmitted through air. Cannot be transmitted through food. So Ebola is ruled out in the first one. Right? And what is said here? 2 to 21 days is the time period for symptoms to appear in. right? And 50 to 90% people infected with this virus die. High rate of death, high mortality rate. Then what comes? No specific treatment for the disease. We don't have any vaccine and all this. Where vaccine works, where the virus is stable, something is there. But there are certain viruses which change. Today you can prevent from getting by disease, but you cannot be reassured the next time there will be same one. You mutated, something else will be there. And that is the case in case swine flu, bird flu, all this flu viruses. They keep on changing. Right, this part is there. So what is challenge today? There are certain diseases where scientists, doctors, they're struggling to get the new type of method for containing them or treating them. So still the vaccines are not there. Malaria, no vaccine is there. Worldwide trial is going on for malarial vaccine. Right. For HIV, no vaccine is there. Again, the scientists are trying to get the HIV vaccine. Then any kind of flu viruses, no vaccines are there. Right. So this part, in this case there, one source you follow that. Another is that disease part and all, that will be, I think, in the general science segment, the teacher will take the biology in this part, will cover there in terms of all the names, factual part, right, which will be given there. But apart from that, from my side, let me do that. I'll compile and put it there, the causative agent and all this part. But when you read the biology part there you will be coming across all the hormones, endocrine glands, then digestive system, human physiology part, there it comes. From the technological point of view, what I will be covering here, so I have covered what biotechnology part. So that is the topic which is there to learn about from technology point of view. At the same time, health aspect, the basic understanding, and the part which comes from current aspect. How you have to link with current aspect, right? So the things I'll put into that. Though in the book this book I have given there, but I'll put into one format: malaria, dengue, chikungunya, so that you know that this current disease for last 3-5 years, which are there, you have the knowledge on that. I'll place that, right? ये basic चीज को याद रखना है, ठीक है? तो ये चीज है, तो ये this one question common, one question you'll find in this areas there, which is asked in the exam. That will be part of it, right? Now I think tomorrow we don't have class. Do we have class? No. I think we are meeting the next week. Next week we have class. So next week I will take the topic of what I was given the list wise there. I think in the list wise after health I have said biotechnology health, I will be going to the area of new technology, renewable energy, new technology in this one. So we'll be learning about nanotechnology, robotics, laser, lidar, all those emerging technologies. That part we'll learn there. And after that we'll go into the IT and computers part. So IT and computers, this will be the next one. And then I'll be going to the renewable energy also there. Right. That part, then I'll say IPR and all this part will cover in that class. Okay? So any doubt you have? I know 3 hours class becomes too torturous, right? And today the biotechnology and all is very torturous topic there. Okay? Interesting also there. Gene therapy and all this part there. वो तो गाने भी अच्छा था, म्यूजिक अच्छा लगा वो, तो ऐसे ही गाने वाला म्यूजिक बजाना पड़ेगा, right? You will get in that. Okay, guys, we'll meet the next week, right? And we'll carry on with the new technology areas there. Okay. So thank you everyone.